Well, thank you, Raja, again for the invitation. Today, I'm gonna spend some time talking about the RNA-Seq pipeline that we have integrated in using the interface for differential gene expression. My name is Luis Santana Quintero. I'm with the Hive team inside CIBER in OBE, Office of Biostatistics and Epidemiology, and the Hive. So let's start with, well, first a disclaimer that this presentation reflects only my views as an author and should not be construed to represent FDA's views or policies. Again, my comments are an informal communication and represent my own best judgment. These comments do not bind or obligate FDA. So now we can start like the outline. This shows different parts of the presentation. I will give a brief introduction to RNA-Seq, RNA-Seq analysis, and bioinformatic components that are divided into three. I will show you how to run the pipeline and get results, and then we're going to talk about data quality control and visualizations. So let's start with the introduction, experimental design. We know that RNA-Seq is a powerful tool for transcriptome profiling, and it allows us to better understand the functional elements of the genome. It helps revealing the molecular components of cells and tissues, and it helps to understand development and disease. It is important to mention that the Hive RNA-Seq pipeline uses a reference genome to align the reads and is optimized for differential gene expression analysis. You can use it to do the analysis among different cell populations. It can help us to find differences on treated versus untreated cell lines, wild type versus knockout, and cell populations from the same tissue at different time, like for example, neonatal versus adult. Important to mention that RNA-Seq can also be used to run de novo transcriptome assembly, but our pipeline is not optimized for that. If you run the transcriptome assembly, you first assemble the transcripts with a de novo algorithm, and then you align those against the genome. And this pipeline usually it helps you to detect de novo transcriptomes, splice variant, and novel gene finding. That is not our pipeline, is not optimized for this one, but you can also do that with RNA-Seq. So for the experimental design, I'm not going to go into much detail. I'm going to focus on the bioinformatics part, but we just have to remember that the goal of the RNA-Seq analysis is to observe a reproducible effect that is due only to the treatment, trying to avoid bias, while we measure the variability of the experiment to estimate the effect when the measurements are repeated with similar samples. This is with the use of replicates. So it becomes really important to perform these experiments with replicates. They will help us to estimate the variance of the experiment, and the statistical tests will make some assumptions about the gene expression differences by removing this bias. So mainly there are two types of replicates. The first one is a technical replicate, where these are repeated measurements of the same sample, either by preparing different libraries, multiplexing the samples, or using different lanes in the flow cell. And the goal is to eliminate random noise associated with protocols or equipment. The other type of the replicates, the biological replicates. These are replicates from biologically different samples. They can be different animals or independent growth of cells or tissue, and the goal is to eliminate random biological variation. If our experiment fails to measure the variability, then we won't be able to generalize our results, and we may present results that will represent only the changes on those 10 mice that we sacrificed on that specific day on that lab by that one person. And that's something that we will be the main reason to avoid. We won't be able to reproduce the results. So that's something that RNA-Seq and the use of replicates help us with that part. Next, I will start talking about the different bioinformatic components of the RNA-Seq analysis. Here, I present the RNA-Seq pipeline that we have, divided in three main components: primary, secondary, tertiary. I will visit each step on the pipeline and give more information on the next slides. But in general, the primary analysis consists of the initial raw data, conversion, demultiplexing, and QC metrics on raw data. The secondary analysis consists of filtering, aligning, quantifying, and normalizing this data. And then the tertiary analysis consists of running the differential gene expression, functional pathway analysis, and now I will start describing each one of them in the next slides more in detail. So number one is base calling and demultiplexing. Usually this is the files that are coming from the sequencing machine, BCL files. These are converted through BCL to FASTQ into FASTQ files. They are being demultiplexed. Sometimes the adapters are being removed. It depends on their settings. And sometimes if you already have FAST or FASTQ files, this step might be unnecessary. But sometimes when you get raw data from the machine, you will have to do this step. So this is number one in the pipeline. Number two, it's a quality filter and profiling. So for this step, we are using a tool, it's called FastP, that allows some quality control of the reads. It produces some reports. It allows us also at the same time to filter those reads. We can filter low complexity, low quality, too many ends, based on the length. We can trim from the front or the tail of the reads, cut low quality bases, adapters, remove them, correct some mismatches based on the overlap regions of the parent reads, trim poly G, poly A from noise from the machines, from the instruments. The tool is able to process or pre-process using UMIs. Not that we're going to use it for the RNA-Seq for this pipeline, but this tool is able to do that. Again, FastP gives us some quality control in a single HTML and report using JSON in case we need it. We can get a summary table including all the statistics for the read quality before and after filtering. I will show you later. But yes, this is basically the description of the tool, like how the tool is like what are the word, like the tool, like features of this tool that we are using. For the quality, like the read mapping, we are using HiSat2, that is a hierarchical indexing for splice alignment of transcript. It's a sequence alignment program. It uses graph FM index that creates the index of the whole genome. Basically, it's a strategy to allow rapid and accurate alignment of the sequencing reads, and it allows sequence alignment on the transcriptome. So as you can see, there are some gaps on the introns that is just aligning the exons, and HiSat2 allows those alignments. We get once we get the BAM file or the SAM file from the HiSat2 alignment, we move to the next step, that is the quantification. For the quantification, we are decided to use feature counts. Feature counts is a tool that helps us to map these sequence reads that already got aligned and assign some genomic features. So basically it quantifies, it counts indels, junctions, it can find structural variants in the reads, it can find multi-mapping and multi-overlapping reads, full exclusion, full count, fractional count. So all these features are there that we can use, and we use it to count. We use it to count based on where it got aligned, if it's to gene A, gene B, multi-map, all these different features that feature counts has, like in order to do the quantification. And step number 5, we are using the multi-QC. Multi-QC generates a report based on multiple samples, and it helps to process QC from generates an HTML report. We can easily see all the samples in one table or in one graph. It gives us information of the GC content, percentage of assign, align, based on the previous results of the previous tools. And we can get bar graphs from feature counts and HiSat2. Aggregated FastP results can also be displayed with multi-QC. So it's already integrated into the pipeline. Then we're moving to the normalization. It's important to mention that normalization removes a specific bias on the sample, on the data. This bias can affect downstream applications. So we need to consider some factors. The ones that we are considering are sequencing depth. Like, it's important because like the gene expression between samples could be that one sample has more sequencing depth like than the others. So in this example, each gene in sample A, it looks like it has double expression as relative to sample B, but this is because sample A has twice as much sequencing depth. So we need to consider sequencing depth gene length, that it's how long is the gene. Sometimes when we only count reads aligned to gene X or gene Y, we count and the count is the same, but the length of the gene has to be considered in order for the normalization. We cannot compare them directly. We have to do some adjustment, and that's through the normalization. RNA composition, this is also important, like it allows accurate comparison of expression between samples. So for example, the counts of an asymmetric gene, it can take up most of the counts for sample A, as you can see, and the rest of the genes appear to be less expressed than those same genes in sample B. And those factors need to be considered. There are multiple metrics for the normalization. I'm here just mentioning some that we are planning to integrate into the pipeline right now. They are not integrated, but those are the metrics that we decided to add probably in the next release. And is the upper quartile, that after removing genes with 0 counts, these genes are divided by the upper quartile of the counts and multiplied by the mean upper quartile across all samples. The trim mean of M values, that's another well-known metric that it's based on the hypothesis that most genes are not differentially expressed. So for each sample, this TMF factor is computed with one sample being considered as a reference and the others are test samples. And it calculates the weighted mean of log ratios between this test and the reference. Usually a value of 1 is expected. If not, then a correction factor must be applied to the library sizes for the normalization. Relative log expression, it's similar to TMM, in which it assumes that the most genes are not differentially expressed, but it's a different metric. It uses a mean of the ratio for each gene, and it reads counts over the geometric mean across all samples. The other one is the gene length corrected TMM, that is very similar to the TMM metric, except that it takes into consideration the gene length to do the normalization. So all these are just some of the normalization methods that are out there. But for our analysis, for the differential expression analysis that we're using DSEQ2, we use the raw counts. DSEQ2 estimates the variance based on the counts, and it tests for the differential expression based on the negative binomial distribution. So again, raw counts are using normalization factors for account for the library depth, estimates the gene-wise dispersion and shrink to generate accurate dispersion to model the counts, and it tries to fit this into the negative binomial model and performing a hypothesis testing using the Wald test. So basically the workflow, it's inside DSEQ2, it's the one that it's there. That first step is to estimate the size factors by getting the median of ratios method, estimates the gene-wise dispersion by because the dispersion reflects the variance in the gene expression for a given mean value. It tries to fit a curve to this gene-wise dispersion. It tries to shrink this gene-wise dispersion to aim to reduce false positives, and at the end it tries to fit it into a generalized linear model for each gene using the negative binomial distribution. The graph on the left displays that the red line is the expected dispersion value for the genes of a specific expression, and each black dot is the gene associated mean, and the maximum likelihood estimation of the dispersion is estimated with the final. So it's adjusted. Those are, I will describe it later, like one of those graphs, we can look at them to run like some quality control checks. After having the list of genes that are up or down regulated, the next step is then step 8, that is the functional pathway analysis. This is performing the list of, based on the list of genes to get some biological insight. So in here, we are trying to determine if there's an enrichment of non-biological functions, interactions, or pathways, try to identify genes in novel pathways, or by grouping the genes together. It uses the global changes to visualize all these genes. So there are multiple tools. This is just an overview. I'm not gonna talk more about the functional pathway analysis today. These are just a list of some tools that you can use. like gene set enrichment analysis, IPA, that is ingenuity pathway analysis, gene ontology enrichment analysis, and KEGG pathway analysis. These are some methods. There are others. But yeah, so that's basically the description of the pipeline. Now I'm going to talk about how to run the Hive RNA-Seq pipeline. Like I will mention how to upload the data, how to submit it, and some results. But first, just a brief introduction of the Hive platform. The Hive platform provides solution for the NGS like data. It can go from the sample to the sequencing, and once the file gets transferred into our database, we can do the computations, the pipeline. We can do the analysis and review, and the goal is to get articles, publications, reports, and meaningful results. And we can do this using the Hive. We have an object-oriented paradigm that helps us to reproduce the experiments and the results. So yes, I'm not going to talk a lot, but this is like an overview of the platform. And how to run the pipeline? Well, first we have to make the data available to the Hive, to the platform. We can upload the data directly to Hive. We click on the uploading icon, we choose the files from your local machine, and we start the upload. The second option, there are many other options, but the second one is to download publicly available data from a repository. In this case, could be a SRA from NCBI directly to the Hive. We go to All Objects portal. We select Other. We select the tool that it's NCBI SRA download. We use the accession numbers to identify which ones do we want to get, and we submit the job. And we forget for some time, and the Hive will be able to get the data. In this case, are FASTQ files from SRA. This is just an example of the screenshot of the SRA. In this case, we are using PRGNA 229998. For the current presentation, we're using data from there. It's a publicly available data, and we generated results using this data. This is a screenshot of the interface of that tool. It's a Hive RNA-Seq. We try to make it as easy as possible. We try to just provide a job name. You select your preloaded sequence reads, even for R1 and R2 for the parent reads. You select your genome, like the index of the genome for the HiSA2 index. We select the respective annotation file, either in GTF or GFF format, and then these feature types, attribute types that feature counts uses to extract the information from the annotation. We know that there has been some, we're trying to make it easier for the researchers, and this is something that we are working to get a next version. It's under development, and trying to just make it simpler, like for the researchers just to input their FASTQ files and just say, I want to get results from the RNA-Seq based on this organism. And we're gonna preset all the other parameters that we were using, like exon, gene ID, ID. If you don't set or we don't set them correctly, we are not gonna get expected results from that pipeline. So this is important, and we're working on that, trying to make it easier. Once the computation finishes after several hours, it depends on the number of samples that you have. You can access the results by clicking, again, could be all objects, computations. You click on your job, and it opens a new a window with the results. For now, we have only available files. The important ones are the feature count table and the summary in the TXT file. We also can see the FASTP results for each sample in HTML and JSON format, and multi-QC aggregated reports in HTML and TXT files are also there, so we can download them and look at them. And then we have a list of all the alignments for all the samples. They are in SAM format and splice site file in TXT. We can download every individual file or the entire results if we click at the top of the page to get the results. And this is how you get results. Now I'm going to talk about what to do with those results. Like, what can we do with the feature counts table? And what we can do, this is again like the pipeline. just as a reminder, what we can do is do some quality control checks. We are interested in sharing the results that we are getting with researchers or collaborators. So for the primary steps, like we need some quality checks, questions to that we can answer. With this one, it's like how many duplicate reads are on my samples? Like how many raw reads each sample has? What is the average quality of the reads? Is there an AT or GC bias? What's the insert size distribution for the parent reads? So the report in the multi-QC, it gives us most of this information. It can, and all the samples are in those graphs. For example, like identification of PCR duplicates. Like we know that the sequence library should contain a random and complete representation of the genome. Most sequences should occur only once. If we have a high duplication rates, it could be an indication that there's some PCR over amplification, and it could be because there's an insufficient initial or starting material. So there are indicatives or indications that we need to communicate to the collaborators to say like something happened with this sample or with a set of samples, with a batch, with a run, and they can also take a closer look to the results. The quality score, using the Phred scores, like as we know, like the plot, this one is based on the quality scores by the sequencing platform. Usually for Illumina sequencing, we know that the reagents degrade through the sequencing run, which is why the last bases tend to get worse results, or at least for the scores. Usually a value of Q30, like values of Phred scores above 30, is considered a good quality score, but we need to just pay a quick, pay attention to this graph. In case you see something unusual, then you communicate those results and then you raise some flags. The GC content, again, is important because it can help you to detect potential AT-GC separation. It can affect gene expression quantification. And what we are expecting is the ratio of GC and AT equal. A large variation error in the first 10 bases on the read number 1 is expected because of the primers, so that considers a bias, but after the 10 bases, usually it should be relatively equal, like that GC content along the samples. Insert site distribution, it's another metric that we can look at it, and what it does, it estimates the inner distance between the two pair RNA reads. So it's the fragment, you have like the two reads coming from each side of the fragment, and there's a gap in between. So this gap should match the gel size selection during the library preparation. If it doesn't, then again, raise some flags because something could be not good. Now we move to the secondary part, secondary analysis, and there are some other quality control checks that we can use. Some of them are how many reads got aligned, how well did these reads align? Also, how many reads were filtered? Do I have enough reads to get meaningful results? Do the reads got correctly assigned to the gene? Like all these questions can be answered with the quality control checks that we have in the same multi-QC report. So for the filter reads, like we are not expecting a lot of the reads being filtered due to low quality, like low complexity, too many ends, the length, too short, poly A, poly G regions. So we can get these graphs and we expect high, usually more than 90%. Some experiments could be up to 80%, but that's already like something that you should pay attention. Like you should raise some flags if there are a lot of filter reads out of the samples. During the alignment, we are trying to get clean reads aligned to the reference genome. HiSat2, again, it's used for the alignment, and this graph shows you a lot of different type of reads that got aligned, like parent map uniquely, like those are the ones that we are looking forward to, like pair reads that are being mapped to a single position of the genome. We can also get statistics for the discordantly uniquely, that those are one pair got aligned to one chromosome and the other one got aligned to a different chromosome. So that's something of concern, because if they come from the same fragment, they should come from aligned to the same chromosome usually. like one made map uniquely, like multi-map, one map. So all these statistics are there for us to take a closer look. In here, you can see that some samples are, they have a low map uniquely, and that's something that this is public data, so I'm not too worried, just for us to present, but this will be something for us to report back. And as you can see, going from the alignment to the quantification, the same samples that have a poor number of aligned reads are reflected in the quantification. So it's something that it affects all the pipeline, and we can detect them earlier, like we will save a lot of time. So for the quantification, again, like we can look at the abundance of transcripts reflects the gene expression level. So the feature counts, we usually work with the assigned reads. And these are the number of reads uniquely assigned to a specific gene to be used for downstream analysis. So those, in this case, the blue ones are going to be the ones that we're going to use for the next step of the differential gene expression. And we at least expect to have at least 10 to 15 million reads. If this is a human sample, that will be the minimum number that we will expect to get a good results for the RNA-Seq. And here you can see all the unassigned reads are not being considered for the rest of the analysis on maps, singleton, chimera. Sometimes we want to look for answers of why this is happening. It's out of the scope for the pipeline, but it's something for us as bioinformaticians to look at these graphs and make some assumptions and communicate those to collaborators to let them know that something is off on certain samples. Again, data quality control statistics, there's a summary. We can download the table from the multi-QC. We can use it for our report, for the publication, to communicate to others, and not only with a graph, we can use it in a table format. Now, the last one is a tertiary analysis, like again, quality control checks, like that we can do, and questions that we can answer is like how the dispersion of each gene based on expression level is represented on the samples, like how well the unsupervised classification performs on the data. Like, are these samples together? Like once we cluster them, are the clustering showing any outliers? How many principal components are needed to maximize the variance on the samples? We may find some bias. If we have group information of the replicates, can we provide a global view of how the genes are differentially expressed? So we can use those to get answers for these questions. For the tertiary analysis, as I mentioned before, we download the table from the Hive computation results, and we use R, like the R scripts that using DSeq2 R package. Again, DSeq2 models the raw counts, and it tries to fit the negative binomial model and performs hypothesis testing using the Wolf test or likelihood ratio test. Some of the graphs that you can generate with these R scripts are scatter plot of normalized counts, dispersion plots, PCA, hierarchical clustering, MA plot and heat maps, p-values, histogram, and volcano plots. So I'm gonna show quickly like some of these graphs. The scatter plot of normalized counts, it's a quick view of how the normalized counts of the top 10 most significant genes by the p-value, adjusted p-value, are between the two conditions. So we will expect them to have similar read counts between replicates. And for these top 10 genes, yes, we see that the control and the treated are all together for these genes. That's something that we expect. If not, then again, something that we may not have a lot of genes that are up or down regulated. So observation is that yes, the top 10 most significant genes are differentially expressed between control versus treated. Dispersion, again, DSeq2 tries to estimate the dispersion, and this is the graph for each gene based on the expression level. Using maximum likelihood estimation, tries to feed a curve, and it tries to adjust the values of the dispersion by doing the shrinking. So this curve is displayed as a red line. This is the estimated data points expect to form a cloud around the curve, and the pattern or curve does not follow the general expectation. We might have a contamination or an outlier sample. In this case, we can see that the data show an expected cloud pattern scattered around the curve, the dispersion is decreasing, and the mean expression levels are increasing. So we are good with this experiment. For the clustering, some of the things that we can get are the PCA and hierarchical clustering. For PCA, it's it tries to find and maximize the variance. So it can be also used for dimensionality reduction, and we will expect the biological replicates to have similar scores. So if not, then again, something to notice. The hierarchical clustering, again, it displays the correlation of the gene expression for all the pairwise combination of the samples in the data sets, and we expect the replicates to be according in the same group for these two graphs. For like MA plots and heat map, the MA plot shows the mean of the normalized count versus the log-to-fold changes for all the genes tested, and usually the part on the top, the red dots are usually upregulated. The red under the zero are the downregulated. The heat map is the representation of the differential expressed genes by adjusted p-value. With red are higher expression values, blue is lower expression values, and we can see a difference among the control groups and the treated groups. So something to share. Also, we can look at the histogram of p-values and Volcano plot. The histogram of p-values gives us a global overview of how the genes are differentially expressed for the comparison. Volcano plot shows the up and down regulated genes based on the adjusted p-value and the log-to-fold change values, and it gives us a clear idea of how many genes are up or down regulated for the experiment. And then that's it. This is just a reminder of the whole pipeline that we have from going to primary, secondary, tertiary, and conclusions. Like, it's important to remember for the experimental design, use replicates to help to capture the variability. The Hive RNA-Seq pipeline, it's optimized for detection of gene expression changes. It allows identification of genes that are mostly affected by different conditions. Data interpretation requires several analysis steps. Be aware of different normalization methods. Be aware that there are data quality control checks that we can use through the pipeline. We can identify issues sooner rather than later that can affect some results, and use the parametric methods based on the Poisson or negative binomial distribution to try to capture both the technical and the biological variability that is on the sample, so we can reproduce those results, or someone else can reproduce those results on a different similar experiment. Doesn't have to be identical. These are just the references of the tools that we have used for FastP, MultiQC, HiSeq2, feature counts, and DSeq2. And finally, some acknowledgements for the from me to the Hive team, FDA Hive, GW Hive. This is an old picture. We need to update the picture. But yes, special thanks for the major contribution for this presentation to Charles Chung. Like, thank you, Charles. Like, it was a very and yeah, questions. I'm happy to answer any questions. I hope. And
let me say thank you very much to Luis for demonstrating how to do RNA-Seq in Hive using third-party tools that I think many people have probably heard of. I want to take just a few minutes here, though, and tell you about another way to do RNA-Seq in Hive using tools that are native to Hive, linear discriminant analysis in Hive. So there's 3 steps to this pipeline. The first is an alignment, and you do this like you would with any other alignment. The native tool for alignments in Hive is hexagon, so you carry out a normal alignment for any of the samples that you want to analyze. The second step is to send it to a tool called Alignment Comparator, which will combine all that data and format it in a way that the RLDA tool can use. And so then once that data is there, you'll use that table as input, but then you also have to tell it what categories you're interested in. So that could be healthy versus tumor, or infected versus not infected, or whatever it is that you're looking for. And then in the last step, you analyze that data using RLDA, and it'll try to find the best way to separate your data into different categories, or different kind of clusters. And so that's it. It's a pretty simple 3-step pipeline, and I'll walk through it very quickly, just in a few minutes. But first, if you're not familiar with linear discriminant analysis, it's over here in the dimensionality reduction family, where principal component analysis is probably the most famous member of this family. And it's one that you've probably heard of. But PCA looks for variance in a sample, and linear discriminant analysis explicitly looks for differences between the samples. So just to illustrate what I mean by that, let's assume that you have two samples and you ran an RNA-Seq experiment on them. There's sample 1 in blue, sample 2 represented by this orange color. So if you're looking at how much of gene A there is, you won't see any significant difference between the two samples because they both have a similar left to right spread and it heavily overlaps. And same with gene B. If you're just looking at gene B, you still probably aren't going to see any significant difference between those two distributions. Each one of those two genes is one dimension in the data. And so if you're looking for how much of gene A there is, or you're only looking for how much of gene B there is, you'll miss the fact that there's this huge gene A by gene B effect. And so that's what RLDA does. It tries to look for some axis that separates that data. So where PCA tries to maximize the variance and it doesn't care what population the sample comes from, RLDA tries to maximize the separation of the data points. So in this example, I'm showing you 2 dimensions because that's easier to visualize that way. It's at least a good place to start, you know. But you could do this with 3-dimensional plane or a 10,000 dimensional manifold. We wouldn't be able to plot that and show you what it looks like because that wouldn't make sense to a human brain to represent it that way, or to try to represent that many dimensions in some sort of graphical representation. But you know, plots are just vehicles to help us understand the data anyway, and so they have a scope of utility like anything else. So anyway, so that's the way this works. It could never do this by brute force calculation, of course. If there's 190,000 transcripts in the human genome and any given cell is expressing, you know, as many as maybe 12,000 of those, then even trying to figure out if there's a 2-gene effect is enormous because you would have to step through every single possible combination, let alone if there was more than 2. And so instead, what RLDA does is it finds the center, well, I should say LDA. What LDA does is it finds the center of mass for each cluster and then compares it to the center of mass for the whole thing, and then also optimizes the scatter for each group. So you optimize the distance from the mean and the scatter for each cluster, meaning you maximize intergroup distance and you minimize intragroup distance. And it does that using covariance matrices in this case. There's a couple of different ways to implement it. And so it doesn't really matter what dimension it's in, you know, it's just looking to separate the clusters. And so that's the basic idea. And so it's probably not helpful to think in terms of linear plots at this point because it's just taking some, you know, some numbers in an arbitrary dimension. But the point is that you can do this with as many dimensions as you want, even if you can't imagine what the physical shape of the boundary of that those dimensions would look like. And that's how LDA works. And this version is, quote, regularized because it does a little bit of subsampling to improve, you know, in large part to improve performance by adding a small value to the data uniformly, 0.01, in fact. And that's because the algorithm can get really bogged down on tiny numbers that go on for lots of decimal places when all you really care about is the first few digits of it. And then even more so if there's a 0 probability in there, which can also really cause some problems. So this is a way of substantially improving performance, and I'll link you to a paper at the very end if you want to see an example of it in action. But the basic idea is you're just looking for features that can distinguish 2 things. And just like PCA, it does that by trying to find a perspective on the data. So, you know, let's say you have two data sets, and they kind of, you know, let's say they look like this in space. They're both going to probably end up showing you some perspective on your data that kind of looks like this, if they're already separated like this. But if the 2 data sets kind of look, you know, let's say like this, where it's partially overlapping, PCA is going to show you, whoops, there we go. PCA is going to show you some perspective on your data that looks like this because it's maximizing the variance on your data. But LDA is going to try and show you a dimension that looks like this because it's showing you how those 2 data are separated. And so this, you know, that's sort of the fundamental. They're both doing dimensionality reduction to try and kind of show you what your data looks like in some other perspective, but they're looking at two different goals. And so for LDA, it makes it a perfect fit for something like RNA-Seq, for example. But I should also mention that it doesn't only have to be used for that. You could theoretically use it for something like SNP detection or something else. But the way that this pipeline is built is specifically designed for RNA-Seq. And so that's what I'll show you. Okay, so for the first example, I should say for the example, the first step of the example, I have 4 samples and I've named them A1 through A4. They're just male healthy, male disease, female disease. And so I just, run a typical hexagon alignment, much as you would in any other one. So you set up your parameters in whatever way is appropriate for your experiment and the species that you're trying to do the alignment for. Of course, just be sure that you're aligning to the appropriate cDNA here in the reference slot, since this is an RNA-Seq experiment. And just a couple of quick things to be aware of. If you see a really high number of unaligned reads, that's probably not good for most experiments, and there might be a problem. I think you can see as high as maybe probably 20% or so, and it could still be okay. Again, depending on what you're doing, because there can be issues with read quality, and some reads get flagged for ambiguity or other reasons. But if it gets up to almost 40% or higher, that might be too high, depending on what you're doing. And then the other thing to be aware of is, or at least checkpoint at this step, is this saturation plot. So the shape of this plot looks pretty good, and you'd hope to see something like this, but you can notice that it's calling, more than 130,000 transcripts here. So if there's roughly 190,000 transcripts in the human genome, that's somewhere in the neighborhood of maybe 70% or so of all transcripts being detected. And so for most cells, you wouldn't expect to see more than probably 12,000 transcripts for any given cell, I would think. Maybe if this is embryonic tissue, or these are cancer cells in culture that have maybe had too many passages, and so they've accumulated a lot of mutations. Or I think the thymus is known to express most of the transcripts in the body, so that it can screen out antibodies that would otherwise cause an autoimmune reaction. I think, or maybe it's just a clinical sample and it wasn't very precise when the tissue was taken, and so there's multiple cell types that are being represented here. So I don't know, there may be some cases where you would expect to see a ridiculously high number of transcripts, but if this is something like a cell culture experiment, particularly a monoculture, where you expect to see one cell type represented, then you should not see numbers this high in most cases. And so this is, again, just something to be aware of. All right, so to aggregate all of those alignments into one, you go to the portal at the top header bar, and then on the right at the top, click on the alignment comparison algorithm in the top right. In the general tab for the tool, you'll just pick the alignments that you want to use. And so I picked all 4 of the ones that I told you about earlier. And then at the advanced tab at the top, you need to give it an annotation file. We've already loaded a few annotation files for a few common species like human and mouse in there. But if you need help finding different species or getting them in there, just let me or Raja or Naila Gulzar know, and we can show you how to do this. The last column in a GTF file says, in that GTF format, will say which features can exist. And so Hive automatically pulls that out and populates the next 2 fields with it. So the collapse hits by and the output IDs. So after you've picked your annotation file, you can come to these 2 fields here and pick what you want. And so I picked transcript ID for the first one, and then told it to output to gene name. And so what I'm saying here is that take all the hits from all these alignments, and anywhere that it aligns to a transcript ID, map that to the gene name. So that's what this tool is doing. It's aggregating all of that data and formatting it in a way that the LDA tool can work with. Okay, so after you run it, you can open up the computation and click on hits up here at the top, and you can see all the hits that were detected. And so since I picked gene name, that's what it's showing me. So in the first column, there's all the gene names that were detected, and then how many for each of the samples in each of the subsequent columns. So this is the kind of data that RLDA can work with. So now we're going to try and collapse these 4 samples by these 33,289 dimensions here, which is represented by each of these rows. So to do that, you go to Available Files, which is down here in the lower left, and then you pick the one that you want and you archive it. So you can either use activity hits or activity RPKM. Either one of those is fine, depending on what you're doing. In this case, I want the normalized version, so I went to activity RPKM. And then you just click on this archive button here, which is the globe with the purple arrow pointing upwards. And so that'll archive it. And so that pulls it out of the computation object in Hive and ingests it as a separate object all by itself so that you can work with it. So that's how you take that data and you prepare it for use. Again, the only other thing that you'll need is to tell it which categories you're interested in. So to do that, I just went to Excel and I built a table like this where I just listed each of my samples. So again, I just had 4 in this case. You'll probably want to have many more than 4. But you just list all of your samples in the first column, and then you tell it which categories you're interested in. And so since I called these male disease, female disease, and so forth, I think health is a good category to look at. So the first column is health, second column is gender. And then I added another one here. I thought maybe resistance would be a good one to look at too. So that's it. You just tell it which categories each sample belongs to that you're interested in looking at, and then you save it as categorization table.csv and upload it to Hive. And that just goes through the normal uploader. If you're not familiar with that, it's just this upload button here, which is directly on the main page. Again, it's just this blue globe with a purple up arrow. And by the way, this is how you'll get all of your data into Hive from other sources as well. You either click on that button and directly upload it from your computer, or you can click, after you click on that button, there's a button in the lower left that says external downloader, and you can use that to import data from other sources. So that's how you get that file up there. So now that we have both of those, we have the data, which comes from the comparison algorithm, and the categories that we're interested in, which comes from that Excel table, the CSV table, I should say. Those are the 2 things that RLDA needs to work. So again, go to the portal and go to Algo RLDA at the top of the classifications list on the left column. And in the general parameters, you just give it those 2 things. So the comparison algorithm made that table for us, and that's our input matrix. And so just be sure to click this checkbox here because just because of the output of that comparison algorithm. And then the categories file is this categorization table. So the data is your input matrix, and then you tell it which categories you want, and that's it. And then you're ready to go. There's nothing else that you need to change. You can come over to the advanced tab and start experimenting with the bootstrapping if you'd like, or anything else really. There's a description of each of these parameters on the side. It's cut off in this image, but feel free to tweak those as you'd like to. This is also where you can switch classifier modes. If you want to try out principal component analysis, you can do that here. But there's nothing else that you need to change here. Those 2 things are all that it really needs, and it should give you a pretty good answer. Okay, so when you're finished, you can check your results and see the list of the most important contributors for class separation, and that's listed up here at the top. And then you can also get a visualization of the first few dimensions down here in the bottom, and you can click and drag and move this around too. So just like with PCA, the first axis is the most important factor, which is the x-axis here, and then the y-axis is second, and second most important, and so on. And so you can see in this data that these 2 clusters that I picked, in this case, health. Yeah, I guess I cut it off here. You do have to pick down here at the bottom which are the columns from your categorization table you want to look at. And so in this one, I picked health. So you can see that it's color coding. One group of them is red, one group of them is blue. And when I look at health, they seem to they do indeed seem to pull into different groups very cleanly along these three axes here. And so then if you look at the top, you can see the top gene, which is HMGN3, which helps to define that first axis and contributes to the class separation by about 0.06802. And then so on. And so this will find anything that a prepackaged tool like DE-Seq will find, but then it may also find more on top of that. And that's because these other tools will find things that are orthogonal to each other. So, or very close to it, meaning independent, basically. So if you move in one dimension, you're not really affecting the other dimension very much. So that would be if they're, 90 degrees to each other. And so they're effectively independent. LDA does require independence, but that's really only for the classifiers. And even there, it's much more robust to violations of that assumption. So it may take a little bit more trial and error to find what you're looking for, but the potential for discovery is very, very high with an analysis like LDA, because most gene transcription is not independent, of course. The transcription of one really cannot safely be assumed to be independent of the transcription of some other random gene, or really any other gene. And so this makes LDA a very powerful tool for it. And then if you want, you can also check the eigenvectors. This will tell you how important it thinks each of these factors are. And so you'll probably expect to see some sort of kind of seashell shape like this, where the top hits that it thinks are the most important start at the top here, and then it kind of spirals inward from there until it comes to, there's a limited number that it'll plot here, because otherwise it'll break the browser, but you'll get many of them. So this can be a very nice figure. And then if you see something like this, that's probably a bad sign. This is saying that it thinks that everything is 100% important, which means that it's probably just finding noise, which could be a good thing. So if you run a negative control, either a biological control or a technical control, like if you added some categories in the categorization table that made sense as a negative control for your experiment, maybe, or something like that, then you might see something like this, and that might be a good thing. Yeah, so that's a very quick run through of the RLDA tool. And I want to quickly thank Vahan Simonian for designing and building this implementation of LDA. Vahan was the director of bioinformatics at the Sieber Center at the FDA, and he's now in the private sector at Emblema. And if you want to see a publication with an example of this in action, you can take a look at this one here that he published. I've linked to it at the bottom here. So feel free to get started using this tool. And if you have any questions, don't hesitate to reach out and contact me or Raja Mazumder or Naila Golzar. I put all of our emails here for you. So feel free to start using the tool. Thanks a lot for your attention.